Hello, today we'll talk about gametogenesis. Gametogenesis, it is the process of formation of gametes. So it's either oogenesis or spermatogenesis, where oogenesis is the formation of the female gamete and the spermatogenesis is the formation of the sperm, the male gamete. So these, both of them start with the oogonium for females and the spermatogonium for males. These are stem cells committed to the formation of gametes. Both stem cells undergo many mitotic division for self-renewal, and then undergo a process of differentiation to form the gametes. For the oogonium, it starts with it begins with the meiotic division one, where the oogonium is differentiated into primary oocyte, which is arrested in the prophase of meiosis 1, and it is present at birth of the female. Soon after that, it is divided unequally to form the first polar body and the secondary oocyte. This secondary oocyte is arrested at the metaphase of meiosis 2 until ovulation. For the meiotic division 2, the secondary oocyte is divided unequally as well to form a second polar body and the oocyte. And in this case, the oocyte is ready for accepting the sperm. For spermatogonium, this stem cell is differentiated to a spermatogonia type A, then followed by amplification division and the clonal expansion to form spermatogonia type B, and then it will be committed to form the pathway that leads to the sperm formation. Soon after differentiation to a primary spermatocyte, which exists in the prophase of meiosis 1, which is, in this case, it's a diploid, have the double set of chromosomes. This, upon the meiosis, is differentiated to a secondary spermatocyte in which there is equal division and the reduction division of the amount of the genetic material. So each of the spermatocytes, the secondary spermatocyte, have the content of the DNA material. Soon after that, it is divided for the second division, for another second division to form spermatids under a process, a very specific process for spermatogenesis, which is called spermiogenesis, in which the spermatids is transformed to the spermatozoa, which is actually the functional sperm. And this functional sperm is mobile, which is different than the oocyte, because the oocyte is a sessile and cannot move. Upon fertilization, the sperm fuses with the ovum and undergo to form a zygote that will undergo a process of embryogenesis to form the embryo. So the process by the gametogenesis, it is the process by which the diploid germ cells, either oogonia or spermatogonia, give rise to a haploid sperms via spermatogenesis or ova via oogenesis. Actually, both of them are divided into three stages: multiplication phase, growth phase, and the maturation phase. In multiplication phase, the germinal epithelial cells divide mitotically, as I said before, to renew itself and make sure that there is always this germinal epithelium is the germinal cell or stem cells are present to give rise to spermatogonia or oogonia. While in growth phase, when each of these tiny cells produced by the multiplication phase, which is these tiny cells, the spermatogonia or the oogonia, it grows large in size, and in this case, it's called primary spermatocyte or the primary oocyte. Finally, the maturation phase, during which the chromosome number is reduced to half its original number through a process of the meiotic division or the reduction division. So So each primary spermatocyte, as we said before, produces 2 secondary spermatocytes, each containing haploid number of chromosomes. Each secondary spermatocytes divide to produce 2 spermatids that, under a series of morphological changes and structural changes through the process of spermiogenesis, will form the mature sperm. And you can see it here. This is a very good example, representative example of how the ovum got attacked by a lot of sperms. In case of the primary oocyte, it divides as well by reduction division, but in this case, it is unequal division, which is not the same like the case in the spermatogenesis. This unequal division resulted that that you will have 2 different cells, because almost all the cytoplasm goes to one cell, which will form the secondary oocyte, whereas the remaining half of nucleus has very little amount of cytoplasm, and this is the first polar body. So actually, it is a reduction division, unequal division, but it is unequal division for the cytoplasm. While the nucleus, it divides equally for each part for the daughter cells to receive half the number of chromosome or half the amount of DNA material. So the first maturation division, which is the first meiotic division, occurs with ovulation in female, which is the discharge of the ovum from the ovary. For the second oocyte to mature, another division is needed, has to take place, and this again is unequal division, resulting in the production of a second polar body. The production of this second polar body takes place when the egg is fertilized by the sperm. Occasionally, the first polar body also divides simultaneously with the second maturation division. But generally speaking, it doesn't matter if the first polar body divides or not. The polar body may be actually considered as unsuccessful ova, because you have already one ova. So generally you have like, although the male, each spermatogonia give 4 gametes, 4 sperms, while the oogonia only give 1 ova and the 3 polar body that doesn't function anymore. The ovum. The ovum majorly have 3 functions to preform: supply the nucleus with half the content of chromosomes for future, supply a nucleus with half the amount of chromosomes for the future embryo, and supply almost all the cytoplasm. And supply as well the food reserve to enable the embryo, because the food reserve is existing inside the cytoplasm of the ova, for the embryo to develop to a stage where it begins to feed, to have feeding by its own self or from exogenous material. Therefore, the animal ova is a cell of gigantic size, or a very big size, compared, of course, to the sperm. While the sperm has to find its ovum, the sperm, to accomplish this, the mature sperm is equipped with organ of locomotion, which is the flagellum, and with high rate of metabolism, so that it can generate energy which is sufficient for the rapid movement of the sperm over relatively a great distance until reaching the ovum. Once in contact with the egg, it must provide another peculiar structure that the sperm possesses, is a lytic substance to dissolve the ovum outer membrane. Additionally, this leads to delivering the nucleus containing the haploid set of chromosomes, 1/2 of the complement of the future embryo, to the ovum. So the sperm is morphologically divided into 4 parts. As you can see from the figure, the first part is the head, and the head consists majorly of two regions, the acrosome and the large nucleus, This acrosome is enclosed in a double-layered membrane, and it needs to be protected, because the acrosome contains a lot of lytic enzymes that helps sperm to penetrate inside the ova. So as long as the acrosome exists inside the sperm, it has to be protected by a double layer in the head cap region. So this region is called the head cap region. And as we said before, it is important for the penetration and activation of the egg, of the ovum or the egg. The second region is the neck, very small region, and it contains centrioles. It contains two types of centriole, the proximal centriole, which is near the nucleus, and the distal centriole, which is near the flagella or the middle piece. The distal centrioles actually forms the attachment to the axial filaments of the sperm tail, while the proximal centriole, as we'll see after that, it is have a key role in the division of the fertilized egg. So the other two parts of the sperm is the middle piece and the tail. And middle piece, you can see from the figure, it's formed entirely of the mitochondria. And of course, this mitochondria, it provides all the energy needed for the sperm function and the energy needed for the relatively long distance of traveling until reaching the ova. While the tail is the axial filament, forms the axial filament, the middle portion of it, form the main element of the sperm. And of course, it provides motility for the sperm through whipping of the tail. So what about the spermiogenesis? Spermiogenesis, as we said before, it is defined as the transformation of the spermatids into mature sperm. So initially, the spermatid possesses a spherical nucleus, a clearly visible Golgi zone, and a large number of mitochondria. The acrosomic granules is bounded by densely stained membrane visible in the Golgi zone, and it is spread over the surface of the nucleus, forming head cap. The centriole migrate to the pole of the nucleus opposite to the head cap and give rise to the flagella. The mitochondria moves toward the proximal part of the flagellum and arrange it in a collar-like fashion around it in the region of the middle piece. At the end of the spermiogenesis, the not utilized sperm is cast off and disintegrate. So we can see these morphological and structural modification in the following figure. Again, approximately, the spermiogenesis occurs within 24 days. And you can see here the spermatids have the acrosome vesicle bounded by densely stained material near the Golgi region. And you can see the centriole here is located far away. The start of the morphological change occurs by spreading of the acrosome vesicle, which is the head cap, over the nucleus. And the centriole start to move a little bit toward the center. Here, the shape of the shape of the spermatids starts to change, and the microtubule with the flagellum start to arise, which is attached to the centriole, and the head cap, which contains the acrosome vesicle, spread over and over the nucleus, trying to cover the nucleus. Soon after that, the flagellum is being elongated, and the mitochondria start to migrate toward the middle piece of the sperm, and as well the sperm change dramatically in shape to form the head of the sperm. After that, the mitochondria managed to arrange itself along the middle piece of the sperm, and the cytoplasm try to form the excess part of cytoplasm try to be cast off and degenerates. Soon after that, you can see that the acrosome have been concentrated in the anterior part of the sperm of the head region and in the head cap. And now the centriole is attached, the distal part of the centriole is attached to the axial filament or the tail. And soon after the casting off of the cytoplasm, you can see that the whole sperm now is functioned with the main regions of the sperm. And here you can see a real image, a real scanning image of the shape of the human sperm. So to look closely into the structure of the sperm under the electron microscope, you can see obviously the nucleus, which occupy most of the head region of the sperm, covered by the head cap, which contain the acrosome, and you can see the neck with the centrioles connected to the axial filament of the tail. As well, you can see the mitochondria arranged in a collar-like fashion around the middle piece of the sperm. You can see as well there is a few amount of cytoplasm have not been cast off. For the structure of the ovum under the electron microscope, you can see that it has a large nucleus, haploid nucleus as well, like the sperm. And this haploid nucleus surrounded by a huge amount of cytoplasm that is full with some of the mitochondria, and all these granules considered to be yolk granules, or the nourishment material for the future developing embryo. You can see that it's a cytoplasm even distributed into cortical cytoplasm, which is less dense than the inner regions of the cytoplasm. So this is like a comparison between the process of female oogenesis and the male spermatogenesis. In oogenesis, the meiosis initiated once in a finite population of cells. So here there is a finite population of cells. So all females have only a certain number of ova, while in male meiosis initiated continuously in a mitotically dividing stem cells. The oogenesis produce one gamete per meiosis, while in spermatogenesis it produces 4 gametes. The completion of meiosis is delayed for months or years. It's never completed because, as we said before, the ova is still the secondary oocyte waits until the fertilized and just before fertilization to change into ova, while in spermatogenesis, in meiosis completed in days or weeks. Meiosis is arrested, as I've just said, meiosis is arrested after the first semiotic prophase and reinitiated in a small population of cells. And this is very crucial to preserve the finite number of oogonia present in the female. While in spermatogenesis, meiosis and the differentiation to the sperm proceed continuously without cell cycle arrest. The differentiation of gametes in oogenesis occur while diploid in first semiotic prophase, while the differentiation of gamete occur while haploid after meiosis end, which means this one occurs, this differentiation from the spermatids into the sperm. In oogenesis, all chromosomes exhibit equivalent transcription and recombination during meiotic prophase, while in spermatogenesis, sex chromosomes are excluded from recombination and transcription during first meiotic prophase. And this one, this comparison, the last comparison, is very important because, as you know, in females, the sex chromosomes are XX. So it is convincible to have recombination between the two X chromosomes. While in male, the sex chromosome is XY. So if there is a recombination occur between these sex chromosomes, it will distort the genetic material. So that's why sex chromosomes during meiosis in spermatogenesis, in the process of spermatogenesis, doesn't undergo recombination. Otherwise, regions of the Y chromosomes will be translocated into X chromosomes, and regions of the X chromosomes will be translocated into the Y chromosomes. So this figure shows the location of the elements of the spermatogenesis in mammals. So you can see here, this is the structure of the testis, which is where the sperms are collected in the epididymis. Soon after that, the sperm is passed to the outside via the vast difference. If you enlarge a seminiferous tubule, which is the main tubule inside the testis, you can see in the cross section of the seminiferous tubules all the stages of spermatogenesis where sperms, the functional mature sperms, are located in the lumen. If you enlarge further the wall of the seminiferous tubules, you can see that the presence of type A, there is two forms of type A, type A1 spermatogonia and type A2 spermatogonia, and type B spermatogonia. Soon after that, it will differentiate into primary spermatocyte. After the first somatic division, it will go into secondary spermatocyte that give rise to spermatids. You can see this spermatid with some amount of cytoplasm that soon after the casting off of the cytoplasm, you can see the function sperm in the lumen. This sperm is nourished by and supported by the Sertoli cells. So here you can see that the human male can ejaculate over than 200 to 300 million sperm during coitus. And you can see that normally 60% of the sperm must have a normal shape. That's a normal parameter. 60% is accepted as a minimum, while 40% must show vigorous motility. So to be normal, the male normal, it needs at least to have 60% of sperm have a normal appearance and 40% must show a vigorous motility. So you can see here, this is the normal form, normal oval form of the human sperm, and these all are cases of abnormal shape of the sperm. You can see that these sperm have a larger head. You can see these sperm have a smaller head. You can see these sperm have two heads. That means it's not a powerful sperm. It's abnormal sperm. These sperm have two tails. These sperm have distorted head, and these sperm have a shorter or bent tail. All these forms are abnormal. And this concludes our second lecture.